Hello, everyone, and welcome to a session from New Brain Nutrition and Eat to Be Nice. My name is Jonathan Marks, the host, and with us today is Dr. Alejandro Arias Vasquez. He's at Radboud University Medical Center in the departments of psychiatry and human genetics. He's going to be talking to us today about the gut-brain axis and neurodevelopmental disorders. Take it away, Alejandro.
Thank you very much, John. Yes, today I'm going to talk about one of the most interesting subjects to deal with in my scientific career, which is the effect microorganisms in your gut have in the way we behave. This is within the context of our European project, Eat to Be Nice, which you can learn all about it in our New Brain Nutrition website. So let me ask you something. Have you ever felt butterflies in your stomach? This is pretty much a common feeling that we all have, right? And we feel this in many situations. When we're happy, when we're anxious, when we love somebody or we think we love somebody, when we are just up to do something very important in our lives. And perhaps an easier way to understand this feeling of butterflies in your stomach is to realize that this is a very normal response that we call stress. Stress is just a mechanism that our body has in order to tell us to be alert. Okay? Now, we cannot not abuse stress. Otherwise, bad things can happen. But it's just a very simple illustration to make you realize that our brain is communicating with every system in our body all the time. Now, let's put this in perspective a little bit better. Now, one of the most important communication that our brain has with other systems is with our gut. Now, this network is called the gut-brain axis. It is very important. First of all, it is a bidirectional communication. So our brain and our gut are telling things to each other. I will give you a couple of examples. First, think that you have to do something that makes you feel very nervous, very anxious. You have a work interview, you have to meet your boss, you have to do a difficult exam, you're going to ask for a loan, these kind of things. You're very stressed. Where do you feel that stress? You feel that stress in your gut. And that is very typical. And this is going back to this initial slide where you felt, butterflies in your stomach. You feel also stress in your body. That stress is generated in our brain. Now, the other way, so from the gut to the brain, is slightly easier. Perhaps if you eat something that is rotten, you eat some bad food. Now, the nausea that you feel it in your head. So your gut is telling your brain something is wrong down here. Do something about it. And this bidirectional communication, first of all, is very strong. We have hundreds of millions of neurons in our gut or our enteric nervous system that send very fast, very strong signals to our brain at milliseconds. Now, top down, so brain to gut, it's also very fast, of course, because our systems need to inform each other as soon as the stimuli are presented. Now, current research is now telling us that there is an environmental modifier of this relationship. This is a very important thing to realize, that this communication is also modified by other players in our environment. The one we're going to talk about today is the gut bacteria. Now, these are good bacteria. I will explain a little bit more about them in just a minute. But one thing that you need to realize is that these bacteria are good for us. They perform vital roles in our daily life, and they help us actually survive in the world because they have co-evolved with us as a species, and they actually are able to help us process and digest, quote unquote, food that we cannot deal with. Now, some generalities about this. First of all, we are full of them. We're covered by them. They're on our skin, in our mouth, inside our body. They are part of us. They're called a meta-organism. They are called the other genome. If you think about all the genes that are inside each of these bugs, well, they make a fundamental part of our existence. And there are a lot of them. I mean, there are as many bacteria in your body as your own cells. Most of them are localized in our gut, but they are present, as I just said, all over the place. There are many, many thousands of species, and this is a rough estimate as we haven't been able yet to identify them all. And across all these bugs, across all these organisms, you can find around 100 times more genes than we have in our own genome. There are a lot of them, so much that you can account between 1 and 2 kilograms of your total weight to these bacteria. And the first infection or the first time you are in contact with this bacteria is where you were born. And this is very important because depending on the way you were born, that will also, let's say, mark the type of infection you would get. So these are some very interesting factoids about the gut bacteria. And let's go a little bit deeper into what they do. Before we do that, this is a cross-section of a mouse intestine in which you can see the gut bacteria, which is in the top left side of the slide. Then the green wall, the green barrier is the intestine. Then the blue side at the bottom right is what is called lumen of the intestine with some, intestinal cells. So I added this here because it's a very impressive photo showing the complexity of this meta-organism and how intricate the study of this thing is. I will go into some detail into what the things that we do to understand the role of these bacteria in our general, health. Hopefully, I will be able to explain to you why these bacteria are relevant for our brain.
Alejandro, can I ask a question? Do the blue cells migrate past the colon into the gut, and do the microbiota migrate the other way back into the blue area?
So what we know is that there is an exchange of information between the two sides of the barrier. This through specific signaling systems that feed each other information, but also feed each other substances. Mm-hmm. So the bacteria produce specific substances, and I will explain that a little bit later, that are important to us, and they go through this wall. Now, there is very exciting research showing that we can find this kind of bacteria in amniotic fluid during pregnancy, but there's some indication that some of these bacteria might be even present in the brain or inside our cranium. So it's not completely clear that they are, separate or divided. What is known is that there is a very strong communication between our body and the bacteria, and it takes advantage of the permeability of the gut barrier. So moving on, I will try to explain to you how would this work. How would these bacteria that live in our gut would have an effect on our brain through this gut-brain axis. So we saw this already. We know it's bidirectional, and we know that the bacteria affect this communication. But how? So there are three main avenues. And I speak about main avenues because these are the ones that we understand better. That does not preclude, of course, the presence of other avenues of communication or other variations within these three that we haven't yet discovered. So the first one is through the nervous system. Think about it, it's through our hardware. So it's basically the signals that come from our brain down to the gut. They do so very fast through our nerves, but the opposite is also true. The bacteria generate specific substances that can modulate the speed or the strength or the information or the message itself that comes from the gut to the brain. And this is through the hardware, through the nerves. A clear example of this is the vagus nerve. Now, second is the endocrine system. Basically, the way we have to produce hormones and other specific substances coming from, our cells, specific cells. Now, the bacteria, they produce also specific metabolites, specific substances only they can make, but we need in order to process specific, molecules that come from diet. But they also produce substances that interact with our own metabolites, with our own hormones, and actually help us regulate things like appetite or even mood. So the production of these substances is quite important. So keeping these bacteria happy is also something that we need to worry about or be conscious that we need to do. Now, the third one is the immune system. Remember that I told you that you were infected at birth? Now, this is very important because once you get exposed to these bacteria, not only exposed to air and water and, this new world that's outside your womb, your body starts to learn who are the nice guys and who are the pathogens. This bacteria that you get starts training your immune system, telling it, look, I am the good guys. I am here to help you. I am here to make all these nice things for you. So you need to let me in, and I will teach you a few tricks on what you should identify as a bad guy. Now, our own immune system also trains itself because we have special cells for that. But very importantly is that the microbiota helps or integrates itself in one of our very important systems as the immune system. Now, all of these avenues of communication go directly to the brain. So they also affect other parts of the body. But the point I want to make here is that this signaling from the gut goes directly to the brain. Importantly, the signaling from the brain also goes to the gut and affects the homeostasis, or let's say, the way of life of these bacteria in the gut. Now, this is an example of the type of substances and the ways of communication that I was talking about previously. So at the bottom of the slide, you see the gut bacteria. Above them, you see the cell wall of our gut. Now, and above that, you see the different products of the metabolism of the bacteria and the way they communicate to the brain. To the right at the bottom, just above the gut barrier, is something called the SCFA. This is the acronym for short-chain fatty acids. And this is an example of a substance that we humans cannot produce, but that we need for our own, let's say, survival. This substance is produced by the gut bacteria. So we need it. So this is an example. Now, to the middle of the picture, you see a line connecting, the gut barrier and the brain. This connection, this physical connection, is what I was trying to explain before through this nerve communication. It's actually hardware where the gut and the brain are physically connected. Now, there are other mechanical, let's say, ways that the gut bacteria can affect the overall system, including the brain, and it's referring to your question, Jonathan, in which the gut barrier can open up and, let specific substances go through. Now, we don't want that to work, let's say, in an unstructured way. It's having that, what is called a leaky gut, can allow, unwanted substances to go inside the gut. So it's a very complicated way, but it still can happen. Now, this leaky gut has been shown to be associated, with high stress, with some mood disorders, depression even. So it's not something that you should take lightly. Now, from the top-down type of communication, one of the main highways of communication is called the HPA axis. It's basically an axis that determines our stress reactivity. So how good are we to handle stress once we're presented with it? And what happens to our body once the stress mechanisms are, triggered? This HPA axis goes from the brain down and has the ability to actually modify the way our gut bacteria respond to those stimuli. So it's very important to think in the following terms. So if I am good to, let's say, cope with stress, it could be because I have bacteria profile that makes me stronger for it, which is very important. Why? Because we can change actually the relative composition of our bacteria. And how do we change it? Well, we change it with diet. Diet is the single most important predictor of change in the gut microbiota. So to give you an example, at birth, you start receiving breast milk. And you receive breast milk for, on average, 6 months, right? The gut microbiota profile when you're receiving breast milk is one that changes significantly once the baby starts receiving solid foods. Why? Because the type of nutrients and food molecules that you're consuming will promote the growth of specific type of bacteria and perhaps not promote it for other types of bacteria. And those changes will reflect into biological changes in the whole body.
I'm sorry, Alejandro, you just said something before that really made me curious. Are you saying that the food we eat or that whatever we intake, whether it's prebiotics, probiotics, good diet, can affect how we react to stress?
No, what I'm saying is that opens that question. So there is no hardcore scientific proof that things like probiotics or prebiotics make you, or any dietary, let's say, intervention for that matter, that make you
more
resistant to stress.
I see.
That does not exist.
I see.
Now, but that would be really cool to try to research, I think. Mm-hmm. Why? Because we have a potential mechanism to try to answer that question. And the fact with the gut microbiota is that you can change it, you can modify it. You can modify the proportional amounts of bacteria and bacteria colonies that live in your gut with the diet you consume. So a veggie diet will have an associated bacterial profile that will be different for a fat-rich diet. So the idea would be, can we find some sort of pattern, for example, associated with the ability to cope with stress? And then perhaps we can ask people, well, perhaps we should try to imitate that pattern. And if you eat specific types of food or specific combinations of food, you could modify your own bacterial pattern and change it slowly into that of an individual that could cope with stress better. Now, there are two things to remember when you think about these kind of ideas. One, that this is a very long process. So if we want to do this, this is something that happens in a long period of time. This is not something that happens from one day to another, right? And the second thing is that the effect eating food will have in your, in the way your brain works is very small effect. It cannot be a big effect. Imagine if you live in more or less temperate climate with a specific diet. Imagine that you go to live to the north of Finland, where, climate is very cold and the diet is completely different. Now, or you go there for holidays, for example. Then eating that food and staying there for a few weeks will not change your ability to cope with things like stress or your risk for a mental issue. So the effects are very small. And that is a very important realization that people sometimes do not make and that we like to remind people about. We're talking about small effects. Now, we're talking about stress and we're talking about, effects at the brain level. And that is something that we're studying here. And our study goal is to try to understand something related with stress, but not quite. Our goal is to try to understand a little bit more about neurodevelopment. So neurodevelopment is important because it's a condition that we need to have to, reduce the risk for things like ADHD or autism spectrum disorder, some types of intellectual disability. So neurodevelopment is very important. Neurodevelopment starts, of course, in utero. Neurodevelopment not only is related to these syndromes I just described, but it's also related to other, let's say, behavioral profiles. So ideally, what I'm trying to say is that we want to look how the brain develops in order to see if we can, by using, environmental exposures or information from our environment, try to improve any problem that might happen at that stage in life. So neurodevelopment is a complicated construct, and it's based on three main, let's say, pillars. The first one, behavior, the second one, the brain, and the third is genetics. So neurodevelopment, you can see it through behavior of the other person. Now, neurodevelopment is located mainly in our brain. And I say mainly, and that will become clear a bit later, but it's located in our brain. And the instructions on how neurodevelopment works are given by our genes. And this has been proven many times across, many years of research on this. So we're pretty sure about these three pillars, explaining neurodevelopment. Now, in context of, let's say, disease, neurodevelopment can be understood as something that I think many people are familiar with, that is ADHD and ASD, autism. These are two very common neurodevelopmental syndromes, and they have quite a societal impact. It's important to understand neurodevelopment so we can translate that knowledge into improving the quality of life with people that are affected by these two syndromes. So I focus basically on these two, because of the fact that they are very common. They are the most common neurodevelopmental syndrome. Perhaps the most important thing is the fact that they are biologically and epidemiologically overlapping. They present themselves together very frequently, meaning that when a person is diagnosed with ADHD, the most frequent comorbidity is autism spectrum disorder. And for autism spectrum disorder, ADHD is one of the most frequent comorbidities, also presenting in there. So that means that you could understand this relationship between these two syndromes as a continuum. So you start in one, let's say you start in ADHD, but you also share, some traits, some behavioral traits with autism spectrum disorder. And these behavioral traits that are shared, form this continuum. Either you are more towards an ADHD side, you are a more combined ADHD autism, or you are more towards an autism side. Now, the other direction is also possible, of course. If the first diagnosis is autism, you will have a more autistic type of profile, or a more combined, or something that is more towards an ADHD. It sounds a little bit counterintuitive if you're diagnosed with one, that you show a profile more of the other. But remember that I'm talking about behavior. So clinical diagnosis includes behavior, but it's not only about behavior. Now, there are a few problems with, this neurodevelopmental syndrome, right? As I said, it's very common. The prevalence is very high. And of course, that's a societal issue. Now, for us that we are researching this, but also for the clinicians that are treating people, the biology of these entities is very complex. These are very complicated constructs. Now, clinically, we have done a huge effort to try to characterize them properly, but we're still far away. So just to tell you an example, we don't have biomarker for these diseases. So what does that mean? So if somebody wants to know if you have cardiovascular disease, you get a little bit of blood from you, right? They measure specific things, and the doctor tells you, oh yeah, look, this enzyme is elevated, this other thing is elevated, and this other thing is elevated. That means that you have cardiovascular disease. We don't have a kind of information available yet for this kind of clinical construct, which makes it quite difficult to research. Now, and the third problem, in my opinion, is that we're managing symptoms. What does that mean? That we're giving people, medication, which works very well in many cases, but to manage symptoms, but not to cure anything. So, of course, people that decide not to, take the medication, of course, they will have, extra levels of difficulty to handle some matters of, life, like finding a job, getting a partner, etc., etc. These are real complicated issues that make the research for, biomarkers or ways to help, the quality of life of people diagnosed with these diseases better. And that's what we're working on. And that's what has brought us to this, idea of the gut bacteria. So how does all of this actually comes together? So we know that, as I just told you, the diet is one of the, most important predictors of the structure of the microbiota, right? We know that is well known, that is well investigated, that is very strong and very robust. Now, one thing I haven't told you, but now I will tell you, is that diet seems to be one of the relevant non-pharmacological interventions that can help to reduce disease symptomatology. This is quite an important finding, and this is supported by a series of meta-analyses that actually have, researched on what other alternatives are there to treat people with ADHD. Asking parents to give drugs to their children is something that is difficult in some instances for the parents. Now, drugs used for ADHD, they work very well. The response rate is very high, but still there are some concerns about giving medication to children by the parents. In terms of adults with ADHD, many of them decide not to use medication because, of course, the medication will modify some things in their behavior. And people say, well, perhaps I shouldn't take it. I can cope with this. I can deal with this, etc., etc. Now, that is, of course, a risky decision. Now, I am a researcher. I'm not a clinician. So my interest is try to contribute to the quality of life of, the patients via the research of alternatives that could help in combination with the pharmacological treatment, improve the quality of life of the patient. Our idea is that perhaps if we find a modulator of, the behavior, perhaps we could help people reduce the amount of medication they need to use if they can complement this with something that they can use in their normal life. Since we know that diet has an effect on the microbiota, and we know that diet could have an important effect on reducing symptoms in ADHD, we want to investigate the relationship within the microbiota and the neurodevelopmental. Now, all the background I told you about the gut-brain axis is the biology that we need to know and we need to understand in order to answer this question. We know that there is a link between the gut. We know that the bacteria produces specific substances that act at the brain level. So we want to check, that's what we're working on. How can we connect this to the bacteria in the gut and the brain of people with neurodevelopmental syndromes? And find a modulator, in this case diet, to help us improve or change the profile of people with ADHD, perhaps change it in such a way that the symptomatology of the disease decreases. This, in combination with the medication, of course, will improve the quality of life. And that is our aim. In that way, we are convinced we can help patients and their families.
So, Alejandro, if I can just summarize my understanding, we have evidence that the diet does affect symptoms of ADHD and ASD. We know also that diet affects intestinal microbiota. And so now what we're researching is somehow that, the diet has to be moderated through something in the body, and that's the microbiota. So we're trying to understand what microbiota are associated with different symptoms of ADHD and ASD. Is that what we're looking for?
That is the main question, yes.
Got it.
Now, and we formulate this question based on the biological knowledge we have on how the gut microbiota works, or some of the information we have about how it works, of course.
Right.
So this is a very complicated, meta-organism, and the information we have is limited, of course. Now, full disclosure, I am a biologist with experience in genetics. I am not a microbiologist. So I look at this problem from a different angle. But going back to your question, yes, we want to try to answer this question: can the metabolic product of the bacteria work in such a way that can ameliorate symptomatology of neurodevelopmental diseases, in this case ADHD and ASD?
Right, got it.
And this is modulated, of course, by diet.
Very exciting.
Thank you. So in order to work on this, we have some ideas of what we want to do. We ask a couple of questions, big questions that we're working on, to see if we can come up with this solution, or at least part of it. The first big question that we have is there a pattern, of gut microbiota that helps us identify patients with ADHD, or they're like a neurodevelopmental microbiota, right? Some sort of marker. Think about it in that way. Second, if there is, what is it all about? What are the bacteria types? What are the culprits? What is causing it? In order to try to investigate, identify what are these bacteria doing? What type of molecules are they metabolizing? How much of it? How frequent? Can we change it? How can we change it? So the first question is, what is going on there? What is there? And second, how does it work?
And Alejandro, in the first question, you talked before about biomarkers that are available for heart disease by taking a blood sample. If there is a pattern, if the answer to question one is a yes, then are you saying that there could be biomarkers in the stool of people with ADHD that would be able to say, yes, you have ADHD?
In principle, yes, but we're still a little bit far away from that.
Got it.
As you say it, the way to collect this, and I will go into a little bit of detail in that a few minutes later, is through stool. But the idea is to be able to say that there is a bacterial pattern shared across people diagnosed with ADHD or ASD. And that would be a type of marker, yes. Ideally, we want to know what they're doing to see if we can find other types of markers related to it, right, that we produce or that we as hosts have. So just to briefly tell you a little bit of what we're doing in order to answer this question one, we look at what we call the tree of life. Tree of life is just a way to organize or to catalog every living thing in the planet. And this is done based on physical characteristics of this living organism. That can be physical, that could be genetic, etc., etc. And the idea of this tree of life is that you start at the top with very general characteristics, and as you go down through the tree, you get more complicated, more nuanced characteristics that can actually differentiate between different species or subspecies or varieties, etc., etc. So you can really go into high resolution, high detail between living things on the planet. And we apply this organization because what we're looking at from the bacteria are their genes. So we are not looking about the thickness of the bacterial wall, or we're not looking about a physical characteristic, but we're looking at a genetic sequence or a genetic characteristic. So in order to do that, we need to go deep into the tree. So just to give you an example, we start from this category called phylum, which is the first category in which we can separate bacteria from other living organisms and which type of big bacterial groups. And we go down into the tree, and we could go all the way to species and subspecies. But this is very, very difficult, because in order to determine properly the difference between species, you need to do very complicated, very expensive experiments in which you sequence the genome, the complete genome of the bugs. And you need to actually decode that information. That it's very, very expensive. So we don't use the species level in our research, not yet. We use one step higher, that is called genus, in which species are grouped. And at this level, we have enough information that we can find differences in things like, for example, specific production of metabolites or other biochemical properties between bacteria. This is not the highest resolution, but it's a very effective resolution to actually find if there are, specific patterns shared between groups. Now, this tree of life can be used, for example, and I will show you a cartoon about that, in order to compare quantities of bacteria. In this slide, I will talk about, the first level of complexity that we use, the phylum level, in which you can actually identify, the major groups of bacteria that are present in a group of samples, in a group of individuals, humans, animals, etc. Why do we do that at this high level? Well, basically because there is a pattern of sharing across humans. And these two names that are on the top of the slide that sound very funny, these are the names of the two most common bacteria groups that are present in humans, the Firmicutes and the Bacteroidetes. They are the most common. So if you're doing this type of research and you're collecting the stool and, identifying which bacteria is there, your expectation immediately is to think, okay, I should see these two groups as being the most frequent. So this helps you, of course, to check how your samples are, in terms of data collection, if everything went okay, the experiments worked, etc., etc. They cannot tell you a lot about disease parameters or behavior parameters because they are very, very general. But for example, in this cartoon, you can see that you can use these measurements to see if there is a global difference between two groups. In this case, I'm using, let's say, controls versus people with ADHD. And you can say, okay, oh, look, one group shows a global difference that is significant in terms of, the ratio between these two bacterial groups. And this is already hinting you, there is a difference overall between the groups. This could be very interesting. Now you can, let's say, decompose the ratio and then look at the different bacterial groups individually. And then you can see, oh, yeah, well, what it seems to be that there is higher amount of Firmicutes in people with ADHD compared to controls, and there is no difference in the Bacteroidetes. So you could say, perhaps I should start looking in the Firmicutes group before I do anything else, because this analysis that I just did is pointing towards that. This is one way to try to understand this data. try to find global differences. And once you see global difference, perhaps choose that what is explaining that difference and continue your research there. That is one way to. Now, once you find these differences, so what do you do with it? How does this work? This is the million-dollar question, of course, because once we, find more information about how this works, then you can actually think about, treatment strategies, intervention. So first you have to find the difference. Then you can start identifying how the difference comes to be. In our particular line of research, we're interested at differences that can explain, specific behavioral patterns or, changes at the brain level, et cetera, et cetera. Now, one way to do this is to use, animal models. And one of the most informative animal models in the world of microbiota research are mice. One of the things that you can do is that you can rear mice germ-free, so basically completely sterile. And then you can transplant the microbiota of the human to the mice and actually try to investigate what is the effect in terms of behavior and the development of the brain. And that's what we did. We took the stool from persons with ADHD and from controls, and we transplanted that into animals. And we compared, their behavior, and we compared their brain development. Now, we tested their behavior in a white box. This is a measure of anxiety for the animals. The anxious animals will not move towards the center of the box or will move less compared to, let's say, healthy, normal, stress-free, animals. What we found was the mice that were infected or were received, the feces from the people with ADHD, they showed a more anxious and frightful behavior. We also looked at their brain, and what we found were very significant differences in brain structure and connectivity pattern, mainly showing reduced volumes and reduced connectivity in those animals that had received the feces from people with ADHD.
And if I may say, Alejandro, that we see those same differences in humans between people without ADHD and people with ADHD. That's been shown.
That's a very good observation. So our group published a little bit over a year ago a very important paper that for the first time described over a very big sample across many centers on the planet, structural brain differences between people diagnosed with ADHD and controls, showing a generalized pattern of reduction in volume in very specific regions of the brain, but also in the global effects of the brain.
Right.
So in some degree, this is mimicking, or because there's quite a difference between men and mice. Of course, you cannot do a direct extrapolation of these kind of findings into the humans. But it's very important to realize that our results were done completely hypothesis free, in the sense that we wanted to actually determine if this intervention, this fecal transplant, would actually generate any type of effect. Remember what I told you before, the effect of this bacteria and the effect of diet, this is very small effect. Now we're seeing that this throughout development can actually produce quite significant differences. So yes, we're working on this very hard, and we're planning to submit this to a scientific journal very, very soon.
Wonderful.
Now, we also wanted to look at humans. As I told you, there's quite a difference between men and mice. And one of the initial questions we had, and we're still working on it, is, can we actually see any differences in actually humans with ADHD and humans without ADHD? We see difference at the brain level directly. So what we did is we compared the way people perform a task in an MRI machine. And the task they had to perform actually indicates one of the, let's say, features of ADHD. And this feature is called reward anticipation. So let me ask you something, Jonathan. If I tell you, what do you prefer? $20 now or $100 in 1 week? What will you choose?
Well, depending on what I want, I would go for the $20 now, but the $100 in a week sounds much better.
So you actually just showed a little bit of ADHD type behavior. So in general, people with ADHD will choose the early reward, and people without ADHD will choose the later reward. And this has been proven, multiple times, behaviorally and in the scanner when people are performing a task. Mm-hmm. And there is a particular region in the brain, and I will show it later, that activates very strongly when people with ADHD are performing this task. So our question was, okay, what if we look at specific, products of the microbiota, specific metabolic products that affect, neurotransmission in the brain that would affect this response. So we know that ADHD is characterized at the brain level to be a dopamine-related issue. So people with ADHD have problems processing dopamine in the brain. So the hypothesis is, okay, what if the precursors of dopamine produced by the bacteria go into the gut, then circulatory system, all the way to the brain through the blood-brain barrier, and then they modify, the kinetics on how these neurotransmitters are produced and used in the brain level, and perhaps increase the risk of ADHD, or on the other hand, protect you against it. Other are possible. So that's our idea. Can we look for bacteria that are producing these substances, these precursors, and see if this bacteria is differently accounted for between, or associated with the performance of a specific task in the machine that we'll be able to see through, an MRI machine. So again, we asked our participants to carry out this reward anticipation task. And then we replicated, of course, this region in the brain that activates. So we knew that our task was performing correctly. We also found that there were significant differences in the way this region in the brain was activated between people with ADHD compared to people without. So also proving that our setup was correct. And then we carried out basically an association to detect if there were any differences in the way, the microbiota was present and reacting or associating with the way these people perform the task. So what we found was that there was a precursor of dopamine, in this case called phenylalanine, that was, let's say, more frequent in people with ADHD compared to people without ADHD. So that gave us quite a lot of confidence. First, that the brain level effects of the bacteria are observable in humans, and that this could be related with something as complicated and complex as ADHD. Now, the future for this is quite exciting, right? We are getting closer to, finding more definite proof about, the presence of a neurodevelopmental microbiome. And this of course, opens this avenue for intervention. This is something that I'm very, very interested in, because we believe that this can help, people in the end. Now, of course, it has mechanistic implications in terms of how this thing works, what type of substances are involved, what other kinds of patterns are there present. We are looking at a very gross, level of resolution. It's a limitation of the method. We realize this problem. So we're working very hard to try to get a lot of information so we can move further towards a more, detailed, more nuanced resolution level.
Can I ask a question there, Alejandro, on point number 2, checking if the gut microbiome might be linked? Don't we already know that food affects symptoms of ADHD and ASD? And if we know that already, doesn't it mean by definition that the microbiome is connected to these same symptoms?
What we have is epidemiological evidence there is a potential effect between diet and ADHD. So we don't have a causal relationship.
I see.
And that is very important, in clinical practice. We don't have a causal relationship, not yet.
Got it. So that's what we're looking for.
We're working very hard to try to determine if that is true. Now, food can affect your well-being, your system in many ways. Of course, directly, because we can digest most of what we eat directly. So we don't need bugs to do it for us. Not right. But of course, they will benefit from, quote unquote, of what we eat. So it's not a direct relationship, because you would not expect people with ADHD to eat essentially different from, for example, the people they share their household with.
Right.
So imagine if you have a family of four, the two parents and the two kids, one of them is, diagnosed with ADHD, the other one is not. But they share the same air, they drink the same water, they eat the same food, basically. So it's not so, direct.
Got it.
So that's why we need to go to a more nuanced level of resolution. Now, the other perspective we have for this research is to try to determine if there's any main way of communication within the gut-brain axis that could explain this. Is this basically because of the metabolites that are produced? So via the endocrine system? Is this some sort of, immunologically related phenomena? So for example, is there some sort of, neuroinflammation, or is this some allergies to specific food items that could exacerbate this behavior? This is very important because this will determine, the course of action. Maybe it's all three of them. Personally, I believe that it's a little bit of all of them. But we still need to have, hardcore proof that this is the case. One of the things that we want to be able to say is what type of food items are, involved in this kind of biological process. Is this something related to the type or amount of bacteria in your gut? So would probiotics or prebiotics be more, efficient or useful in this kind of intervention? Or is just a matter of combining food different, eating less of something and more of something else? This is a very complicated next step that we're also working on with a fantastic team of nutritionists and dietitians from other collaborating centers in Europe. And this is one of the biggest societal impacts that we think we can have with this research, helping people to eat differently. And then if you think about something as specific as ADHD or ASD or neurodevelopment, perhaps we can come up with the suggestions of diet-specific interventions for this kind of clinical diagnosis. And that will help in combination with medication. I need to make that very, very clear, is in combination with medication to ameliorate symptomatology and at least help those patients that are susceptible to these dietary interventions. So we have quite big ambitions for this project. I think we're in the right track. And so far, we're very happy with the results we're getting. And I think, John, that with this high note, I think I finished my talk for today.
Great.
Thank you very much.
Wonderful. Alejandro, this has been great. This is a wonderful introduction to what we're doing with the Eat to Be Nice research, and we are disseminating this on the New Brain Nutrition website. And we're going to have a lot more educational videos coming out. We'll have more on different topics. We'll have a video explaining what Eat to Be Nice is. And I really want to thank you, Alejandro, for this wonderful information and introduction to the research that the Eat to Be Nice project is doing. So this has been Alejandro Arias Vasquez, PhD from Radboud University Medical Center. That's in Nijmegen, the Netherlands. And we're working together as 18 different medical institutions throughout Europe. So it's quite a vast project that's going on. My name is Jonathan Marks. I'm the host for New Brain Nutrition, and we want to thank you very much for attending this session. We hope you'll come back for more. Thanks again.